Welcome to aminacademy.com. In science and technology, let us discuss about mapping of human genome. Gene mapping refers to the process of determining the location of genes in chromosomes. Today, the most efficient approach for gene mapping involves sequencing a genome and then using computer programs to analyze the sequence to identify the location of genes. Scientists are often most interested in the parts of the genome that directly encode proteins. In other words, the protein coding genes. So, oftentimes, there is a priority to identify the location of all those genes in a genome. The process of determining the location of genes in a genome is called gene mapping, as which all forms of genome mapping. The old days involved approaches like sophisticated genetic studies or very tedious process of cloning bits and pieces of a genome. Studying them in the laboratory and figuring out how things were organized relative to one another, including the relative location of genes. But nowadays, such a mapping process usually involves sequencing a genome and analyzing the resulting sequence using computational tools that allow you to identify landmarks of interest, such as genes. So nowadays, most gene mapping efforts first involved sequencing a genome. Now we'll discuss further detail about mapping of human genome. Before that, we'll discuss what is human genome. The human genome is the complete set of genetic instructions encoded in DNA of a human beings. It is organized into 23 pairs of chromosomes with 22 pairs being autosomes, that is shared by both males and females, and one pair being the sex chromosomes, that is XX for females and XY in males. This genome contains all the information needed for an individual to develop and functions, as well as the information passed down through the generations. Now we'll discuss about mapping the human genome. Mapping the human genome refers to identify and recording the location of genes and the distances between them on chromosomes. It's a process that helps researchers navigate the genome, understand its structure, and locate disease-causing genes. The Human Genome Project, a large international effort, mapped and sequenced the human genome for the first time, a significant achievement in scientific history. Key aspects of genome mapping: identifying landmarks. Genome mapping involves identifying key landmarks in the genome, such as short DNA sequences, regulatory sites, and genes themselves. Large linkage analysis. What is that? Early genetic maps were developing using linkage analysis, which examines in the frequency of recombination between genes to determine if they are linked or show independent assortment. Physical maps. Ordered location of DNA clones can be used to create physical maps, which provides a visual representation of the genome structure. Disease gene mapping. Mapping helps pinpointing the location of disease-causing genes on chromosomes, aiding in the identification and understanding of genetic disorders. Disease gene mapping involves pinpointing the location of genes that contribute to the development of diseases, often by tracing genetic inheritance pattern in families or comparing genetic variations between affected and unaffected individuals. This process helps to understand how genes contribute to a disease, allowing for better diagnosis, treatment, and potentially even preventing diseases. Human Genome Project. These projects completed in 2003 produced the first sequence of the human genome, a landmark achievement in genomics. A Human Genome Project (HGP) was a groundbreaking international scientific effort that aimed to map the sequence the entire human genome. This project, which spanned from 1990 to 2003, focused on understanding the genetic blueprint of humans, encompassing all 3.3 billion base pairs in human DNA. The HGP had a profound impact on the field of biology, medicine, and ethics, leading to advancement in disease research, genetic technologies, and our understanding of human biology. Now, different types of genome maps. Linkage maps. These maps show the relative order and approximate spacing of genetic markers on chromosomes useful for locating disease causing genes. Physical maps. These maps show the physical distance between DNA sequence, often based on DNA clone libraries. So far, we discussed a little about human genome mapping. We have done. Please subscribe our YouTube channel.